Oh, morning. Right, thanks very much for those that are here. Apologies for the very dry title of the talk, but it's not going to be, hopefully, as dry as that. So, I work in the wildlife ecology management team, do a lot of work with Osprey around TB freedom, and this is a project that Osprey are sort of funding. There's a few other names there, some familiar to you, Graham Nugent, Jackie Whitford, who aren't here, and Hester Roberts is our genetics kind of expert. I'm more of a statistician, so I'll cover a little bit about genetics, but to be honest, I know nothing about it. It was quite embarrassing, the questions I was asking Hester to try and fill in for this talk, so bear with me. So the motivation for why we want to figure out how many wildlife animals there are in an area, we've got a couple of different areas. So ferret abundance, that's not a ferret obviously, but in most areas where there's wildlife TB, possums are the maintenance host, right? There's been a lot of work done and a lot of questions asked about the role of ferrets of maintaining TB, news articles, research, etc. Ferret here, ferret feeding on a possum, so there's an obvious potential transmission between possums and ferrets. So in Central Otago, there's in some areas, this is Mount Benmore, very few possums and no TB detected in those possums, but still TB in the ferrets. So that's perhaps some indication that ferrets can sustain TB. That's probably got some hidden text. It's not what is ferret. It's sort of the role of what ferret abundance can actually sustain TB. I think some of the pictures have moved slightly with the text here. So the other case that we're working on is pig abundance. So in a lot of areas we use pigs as sentinels for detecting or proving freedom from TB. So if we capture lots of pigs and don't detect it in the pigs, that's some evidence that the area is free of TB. But there is a case for even if TB's gone from pigs, it can sustain in long-lived pigs, deer, and if it pops up again in pigs, that doesn't necessarily mean it's back in the possums. And to get a kind of a sense of the scale of the problem, we need to know what the population size is of pigs. But figuring out the size of a population is really, really hard. How do you know that you've counted all the individuals? And how do you know that you've counted every individual just the once, as this guy's struggling to do? So we can use a lot of field and analytical methods. There's just a range of developed methods in terms of the statistical modelling as well as the field stuff. Mark Recapture is kind of one of the main ones. And there's books, literally, and you can see tagging that goes on these animals to determine individuals. How does it work? Well, it requires 2 field samples, where in our little cartoon example here we catch a number of animals that we know about, we mark them, so bird banding is an obvious one. There's a range of other methods out there. Photo ID can be used for some species, and then you let them go again. And so you know how many you've got out there that are marked. And then you take a second sample and you'll capture a number of animals of which a proportion of those are previously marked. And so if you capture 50% of your sample have got marks, then it means 50% of your population's marked, and you know how many are marked, so you can do a bit of jiggery-pokery and you can figure out the population size estimate based on our 2 samples without having to capture every individual. And there's a whole raft of other methods that get used for that. So it's a pretty common method that gets used in wildlife ecology. The problem is it's really expensive. You have to capture and release animals and capture a second sample. So often we use these indices of abundance, relative abundance, wax tags, cue cards, trap catch, camera indices, things that a lot of you people will be very familiar with. And a higher index hopefully means there's more animals, and if it goes down it means there's fewer animals. But it only gives us a relative abundance. It doesn't tell us actually how many animals there are. And some of those current methods are quite difficult for ferrets and pigs that we're interested in. So we get to the genetic side of it. So close-kin mark recapture is using the mark recapture methodology, but applying it to DNA. And what's nice is that offspring, the little red ones here, carry the marks, genetic marks of their parents in their DNA. And so we get these arrows here, parent-offspring pairs. So if you capture an offspring and an adult and you match them up, you can determine who came from who. And there's a pretty simple equation to say, well, the number of adults in your sample that you capture and the number of juveniles divided by the number of these matches that you get gives you an estimate of the population size. So if you capture everybody, you've got 7 adults and 9 offspring, you end up with an estimate of the 7 adults. So that's kind of trivial, but it works if you only capture a sample of the animal. So you don't have to capture everything. So even if you capture 3 of the adults and a few of the offspring, divided by the number of the matches that you get an estimate that's in the right ballpark for what we're looking at, with uncertainty around it, but we ignore that for this. This is where I come unstuck. Luckily Hester's on board in our project, but Genetics 101, we've got every cell nucleus has a number of chromosomes which have thousands and thousands of genes in them, and all of those genes are made up of these building blocks of, I don't know what they're called, C's and G's and A's and T's, proteins, is that right? I don't know. There we go, sorry. There'll be more people talking later on after me, so this is kind of, yeah. So the different choices for a gene are called the alleles. For example, for eye colour, there's only a certain number of options that we can have in a simplistic term. And what that means is because the offspring inherit those from their adults, so mammals have pairs of these chromosomes, so a mother might have, as a code for a single place where we're looking, an 11 and a 15, and the father might have a 14 and a 16. So the offspring can either have one of those and one of those, or it could have the 11 and the 16, or it could have... So those are the 4 options for that single spot where we're looking, what the offspring could have, ignoring mutations and other things. So what that means is that a parent and an offspring pair, because we're trying to match these up, they'll have at least one allele in common at every place that we look. So here, these 2 individuals, they've got the 15 in common and they've got the 14 in common, so far so good, so we've got a match. We can kind of determine that that's a parent-offspring pair. All right. Problem is you can have matching by chance because there's only certain numbers of options that you can have in each of those places where we're looking. So you could get matches by chance if you don't look in enough places. So even though they matched on the first 2, they didn't match here and so on. So these 2 aren't related in terms of that parent-offspring pair. So the way around that is we look in lots of places to reduce the chance of false matches where they aren't actually a match. The flip side of that is if we look in too many places, we can get false negatives. So we might determine that there's no match, but it could be due to our genotyping, the process that gets used to extract the DNA and create all these numbers, or a number of other reasons. Yeah. So that's where a lot of the hard work can come into it. Other relationships have different types of matching. So parent-offspring pairs will have one in common at each of those places. Full siblings, so where they've got 2 siblings have a shared parent, they'll have one match on 50% of those locations and 2 matches on 25 and no matches on 25. It's just how the numbers work out. And half siblings though will have where they've got one shared parent and perhaps 2 different fathers, so they're half siblings. They'll have a different pattern of the probabilities of these matches, and we can use that to our advantage. There can be complications if we've got multiple generations in our sample, so half siblings will have a similar pattern to grandparents and grandchildren. They'll have this pattern of 50% matching on one location. So what we then need is to get age data and some population model to account for the fact that in our sample we might capture a juvenile, the parent may have died. How do we sort of account for these sorts of things? So as part of the project, we're trialling this method out, ferrets in Central Otago and pigs in the Hohongaroa. We can time the sampling, hopefully, that we only have the juvenile and the adult cohort. So the juveniles are quite still young, and so we can easily determine by their age that these are the juveniles and these are the adults. The animals will get aged as well, so I think jaw bones will be collected as well as the DNA sample. So there's lots of advantages for the ferrets where it may work really well. So there'll be lots of these parent-offspring pairs. We can determine the age structure of adults from the field sample collection. Unfortunately, the inbreeding, so ferrets are really, really closely related. That can make a lot of difficulties in this genetic matching, so there's a few issues to work through still on that. Pigs in the Hohongaroa, slightly different case. There might be undersampling of juveniles, so Osprey would prefer to have longer lived adults as part of their Sentinel program for TB freedom, because if it's longer lived, it's been around longer, so it's had a longer chance to detect TB if it was there. So there's a bit of an issue for us in that. And of course, we might capture a young pig, but the parent may be long dead, and so we can't ever detect it or capture it. So we need to include some population dynamics in there to determine the population size for both of these. So it's still kind of early days, but the method is potentially really, really useful. It's been demonstrated to work, so Mark Bravington from Cyro in Australia has done it for white sharks and bluefin tuna. It's well suited to pest species because probably the bit I didn't mention, which is the main part, is that we can collect our sample just at once from just dead animals. So a poisoning operation or a trapping operation can go out, recover all the carcasses that we can, and do all our matching off that. So we don't need to do a double sample because we can just capture and do the matching from those parent-offspring pairs and other relationships. It's potentially quite cost effective because we can only do this one sample. Mark Recapture traditionally has a risk that you capture a lot of animals and mark them and release them and you never see them again, and if you don't get any matches up, you can't resolve anything out of that. The DNA methodology, from what I understand, is getting better and better and cheaper and more effective, and so that will enable some better matching. Still got these issues of inbreeding to work through, so that's sort of part of the process of the project that we're working on. And there's Grant holding a little ferret there for any questions, so I'll probably race through that a bit quick, but that's all right.
Thanks very much. Yes, any questions?